So in this video, I want to talk about the concept of resting membrane potential, which is basically applying everything that I've discussed in the previous videos about selectively permeable membranes, concentration gradients, gates and pumps found within the plasma membrane. All right, active versus and passive transport. Okay, all of that can be applied to this concept, resting membrane potential. Okay, so basically what resting membrane potential is, in a very general sense, think of it as an electrical charge that the cell carries. Okay, think of it as an electrical charge that a cell carries. So if I was to take a voltmeter and, stick it in the cell, all right, it would come out to, if I was to take a voltmeter and compare the charge between the intracellular fluid and extracellular fluid, I would have some kind of an electrical charge. Okay, so that's what we're saying here. So first, we have to talk about where you're going to find this in the body, and you're going to find it everywhere. Okay, all cells of the human body have a resting membrane potential, meaning all cells have an electrical charge to them. Okay, but some cells in the human body, this is more important for some cells in the body than others. Okay, especially for muscle and nervous tissue. Those are tissues that we call excitable tissues. Okay, meaning that we can irritate them, and they can change their electrical charge, their membrane potential through certain events we're going to talk about, and then as a result, they carry out a certain action. Nerves send electrical impulses down their axons that stimulate the release of neurotransmitters. Okay, muscles contract when you irritate or excite them. All right. Now, all the other cells in the body, like I said, they have a resting membrane potential, but really the purpose of it isn't. I don't know. It's just there. We really don't know exactly why, but it's just created by the concepts that we're going to talk about in a little bit. Okay. And don't worry, we're going to come back to this concept again when we get to our discussion about nervous tissue later on, because to understand how nerves send electrical impulses, you have to understand this first. Okay, so believe me, this is not going to be the last time you're going to see or hear about this concept. All right, so let's get rolling on this. Now, first, before I start talking about this, I want you guys just to take a second and look at this key here. Okay, I'm going to use the color green to represent sodium-specific gates, blue to represent potassium-specific gates, and black to represent sodium-potassium pumps. Remember those active transporters I mentioned a little briefly in the active transport video. Okay, so now you have to bear in mind as well that there's more than sodium and potassium found inside and outside the cell. There are proteins, amino acids, there's glucose, there's many other salts and electrolytes, chloride ions, calcium, magnesium, there's phosphates. Okay, there's lots of other stuff found inside and outside of cells in very different distributions. Okay, but the main focus is going to be on sodium and potassium because these have the greatest influence on membrane potential. All right, so traditionally that's what books tend to focus on, including your own as well. All right, so basically what we have here is, if I have some numbers up here that represent, how much sodium you find outside and inside of the cell. Let's say with potassium. Remember, ECF stands for extracellular fluid, the water outside of the cell, basically the tissue water, and intracellular fluid, the water that's found within the cell. Okay, and what I have up here is 142 milliequivalents of sodium per liter. So think of that, in a general sense, as 142 sodium ions per liter. Okay, in the extracellular fluid, and 10 milliequivalents worth of sodium on the inside of the cell. All right, I'm just gonna, for the sake of saving space, I'm not going to put the units over here by the picture, but you can see them up there. Okay. And then when it comes to potassium, all right, there are 140 milliequivalents of potassium on the inside of the cell, and only, give or take about 4 milliequivalents on the outside in the extracellular fluid. Okay, and I know these numbers are a tad different than your book. I mean, the thing is, you could open 5 different textbooks and get 5 different numbers, but they're all going to fall within these kind of general numbers. Okay. Okay. Now, what we're looking at here is we're looking at a nerve. Okay, I'm using neurons, nerve cells as an example here. All right, so up here you have the nerve cell body, and then you've got a wire-like extension of the cell body called the axon, and then at the very end you have these structures called these pockets called synaptic terminals. Okay, so the cell body receives information. The axon basically, think of this as like a wire where electrical impulses are sent down, and then once that impulse hits these little knobs, that's going to stimulate this nerve to squirt out some neurotransmitters, which you know of as chemical messages that will affect whatever target tissue is this nerve is trying to communicate with, whether it's another nerve or a muscle or a gland or whatever other tissue may be that this nerve is imitating. Okay, so what I did here then is we're looking at this, we're blowing up a section of this axon of this, again, part of the nerve, and we're looking at it up close. Okay, so again, you can see that there is much more sodium outside of the cell versus inside of the cell, and there's much more potassium inside versus outside. Okay, now this cell, this cell is at rest. Okay, this cell is at rest, meaning it's not sending electrical impulses. It's sitting there not being stimulated. Okay, so, and again, within the axon of the cell, there are going to be sodium gates. Okay, there are going to be potassium gates. I should probably have these markers in hand. Yeah, the one I was using. Okay, so there are going to be potassium gates. Okay, and then there are going to be sodium-potassium pumps. All right, so these are found all over the place along the axon. Okay, now remember we talked about the concept of a concentration gradient, right? If you open these gates and there's a difference in the amount of, for example, sodium across the cell membrane, it would naturally diffuse down its concentration gradient until we reach an equilibrium. Same thing with potassium, it would diffuse outward. Okay. Now, what we're looking at here, though, is why, so you should be asking yourself, how come sodium isn't moving into the cell when it's at rest? And the reason why is because when this nerve is resting, the sodium gates remain closed, okay? These gates remain closed, all right, until they are electrically stimulated to open, which we'll talk about much more in depth when we get to the nervous system, all right? But the potassium gates are leaky gates, they're leaky channels, okay? So think of them as gates that are kind of left slightly cracked open. Okay, but remember, these are just proteins that aren't fully coiled up. There's a little bit of space in there, right? Now, you know that, and I'll get to the sodium-potassium pumps in a little bit, all right? Now, you know that, you understand the concept of diffusion, facilitated and passive in simple diffusion, that when this happens, that something is going to move down its concentration gradient from an area of